Welcome, Dr. Goetzman.
All right, thank you. So, as this title suggests, what I'm going to be talking about today is the liver. I'll just start with a few words about myself. As Kira said, I'm a PhD. I work at Children's Hospital in Pittsburgh. I've worked with Jerry Boxley for about 20 years. And while he's seeing patients in the clinic, I'm usually in the basement playing with mice. So what I do is I'm a basic scientist. So today I'm going to be showing you graphs. There's going to be numbers. This is what I do. So I work with mouse models of the various FAODs over the last 20 plus years, and our goal is to try to use these tools to develop new therapies. So today we're going to talk a lot about the liver. I got this list of questions from Stephanie Harry. These are some questions that she says she commonly gets about FAODs and the liver, such as, number one, why is the liver important? How is energy stored and regulated in this organ? Number two, what's the significance of glycogen and ketones? I'm going to spend quite a bit of time on those today. And how does the liver use and disperse these forms of energy? Number three, how does a so-called normal liver function versus someone with an FAOD? Number four, how does the liver function when you're well versus when you might be sick with an infection of some kind or some other illness? And last, how does your liver change as you age? And is the FAOD liver of a child different than that of an FAOD adult? So today I'm going to try to answer these questions, hopefully, and provide some answers to what the liver is doing when it comes to metabolism. So your liver is your largest internal organ. It's about 3.5 pounds in an average adult, kind of roughly the size of a football. And it's a very busy organ. It filters about 1 liter of blood every minute. And the key point I'm going to be making today is that it has many jobs. It detoxifies, it takes things out of the blood, but it also controls the fuel supply for other organs, particularly glucose. So that's kind of what I'm going to emphasize today. So it's kind of logical. You think about the heart, it's a pump, it needs energy to pump, and it uses fatty acids for that energy. So if you have an FAOD, maybe the heart can't pump quite as well. The muscle uses fatty acids, again, as an energy source to fuel movement. And if you have an FAOD, you might get rhabdo. But this list of questions that Stephanie shared with me kind of reveals that there's some fuzziness around what the liver is doing when it comes to fatty acid oxidation and what happens when you have an FAOD. The sort of the key symptom related to the liver in an FAOD would be Hypoketotic Hypoglycemia, which is kind of a mouthful. What this is an unusual form of low blood sugar characterized by both low blood glucose and low blood ketones. And we'll be talking a lot about what ketones are. And getting hypoglycemia is always a concern because your brain needs sugar to survive. So hypoglycemia is a major clinical concern. And this hypoglycemia is usually triggered by something. It's triggered by metabolic stress of some form, typically fasting. This fasting could be induced for a myriad of different reasons. Maybe you're sick with something else. And this fasting state is what calls the liver to action to try to maintain your blood glucose. And if you have an FAOD, maybe you can't do that quite so well. I just want to just remind you of the major energy-consuming organs in your body. The brain comes out at the top. It uses up about 20% of your daily energy. And of importance is, you see those things listed at the bottom. Think of those as like a menu of what these organs want to eat for the day. Your brain can't use fatty acids for energy. It needs glucose. It prefers glucose. If glucose becomes scarce, then it's going to look for ketone bodies. And lactate is sort of a last resort. And the liver can use fatty acids quite well, but usually it prefers glucose if it's available. Similarly, with the bulk of your skeletal muscle is going to prefer glucose. And then you have these two organs at the right, which are actually sort of the exception in the human body in that they prefer fatty acids. Your kidney and your heart, if they had their druthers, would pick fatty acids as their primary food of choice. And if you have an FAOD, you sort of force them to pick something else from this menu. And that all gets exacerbated if you have fasting or some other form of metabolic stress. So the liver plays an absolutely essential role in maintaining your blood glucose levels within a narrow range, what we call homeostasis or a set point. So the top half of this figure, and I don't have a laser pointer, I would show you. You can see there it says number 1, stimulus, and it shows a cupcake. So when you eat a meal, especially something that's high in carbohydrate, you stimulate your pancreas to release insulin. This insulin is a signal to the liver that now blood sugar has risen because all this food you just ate. So the liver wants to bring it back down. So it takes up a bunch of that glucose and it converts it to something called glycogen. And also other organs are taking up glucose along the way, and you end up coming back to your set point. Now at the bottom, you see what happens when you enter a state of fasting. Let's say you miss a meal, you're out hiking and a bear steals your food and you have nothing to eat. Your blood sugar drops. Your pancreas releases something called glucagon. Glucagon tells the liver to do the opposite. It says, okay, now blood sugar is low, you have to bring it back up. So it goes to its sugar bank, which is glycogen, and it releases glucose into the blood to bring you back to your set point. So I mentioned this word glycogen, which is it's a very clever trick that the body has of packing a lot of glucose into a small space. So this thing that looks like a ball on the left, that's a glycogen molecule, and at the center of it is a protein called glycogenin, and it's surrounded by these long branch structures of glucose. And you might be able to pack 30,000 molecules of glucose into one glycogen ball. And typically, between your liver and there's also some glycogen in your muscle, an adult will have about 12 to 24 hours worth of glucose stored up as glycogen in the liver. And that may seem like kind of a broad window, 12 to 24 hours, and that's because it kind of depends on what you're doing. If you're sleeping or you're in bed with an illness, your energy requirements are going to be different and your glycogen will last you longer than if you were, say, that example of you're out hiking on the trail and the bear took all your food and you have to get back to civilization and you're actively moving or hiking, you're going to run through your glycogen much faster. So what happens if you exhaust your glycogen? Well, that's where these two important tricks that the liver has for which fatty acid oxidation plays a role come into play. And these are ketogenesis and gluconeogenesis. You notice they both have this root word genesis, which means create. In one case, the liver is creating ketones. In the second case, the liver is creating glucose to keep you going. Ketogenesis. So let's talk about ketones and show you the chemical structures at the top. Your liver is making these chemicals and secreting them into the blood. And as I mentioned, sort of the major target for that would be your brain. Your brain needs those ketones in the fasted state to keep going. But also your heart and your muscle and your kidney will switch from sugar to ketones in the fasted state. So I love biochemical pathways. That's kind of what I do. So I'm showing here this little circle is supposed to be a mitochondria. Inside that is the pathway from ketones. You have all these different enzymatic steps. These steps are all normal in a patient with an FAOD. Where the trouble comes in is getting the starting material. At the very top, you see those two little chemical structures, and it says 2-acetyl-CoA. That's the starting material that you have to have in order to make ketone bodies. And that comes from fatty acid oxidation. So when you're fasting, your Fat stores release fatty acids. They go into something called the portal vein, travel straight to the liver, and the liver then chops those fatty acids down through the FAO pathway to make these acetyl-CoA molecules, these little short pieces, which can then be used to make the ketones. So if you have an FAOD, essentially what you have is a supply chain problem, where you can't make the ketones because you just can't get the starting material. So we work with various mouse models, as I mentioned, and I just wanted to show you that there is some variability in their ability to make ketones during fasting. So the LCAD knockout mouse, and that's in the upper left, you see these little bar graphs. They have a partial block in FAO because these mice, they're actually a very good model for VLCAD deficiency. So the mice have this second enzyme called LCAD. So these mice still have VLCAD and they have some FAO capacity. And what happens when you fast them overnight, you can see these black bars, their glucose levels are maybe about half of what the wild type, or that's what we call the non-FAOD litter mates. Their blood glucose levels about half of what the wild type mice are. And so are their ketones. Then you look in the middle at those bar graphs, that's a CPT2 deficient mouse. So this mouse has no CPT2 activity at all, zero. So it has essentially a complete block of the FAO pathway. And what you see is if you look at the white bars, that's the wild type mice. When you fast them, their level of ketones, and that's what this graph is showing you, go up about 5 times. Whereas you look at the black bars, which is the CPT2 mice, you fast them and nothing happens. They don't generate any ketones. So this is kind of what happens when you have a complete block of FAO. So every patient and every FAOD will be a little bit different in what their capacity for making ketones would be. So I mentioned this concept of a menu. So what happens is, if you have an FAOD, you've already kind of limited your menu somewhat by taking fatty acids off the table. But now in the fasted state, you're taking ketones off the table. You're forcing a greater reliance on glucose. And so your brain, your heart, your muscle, they're all looking now for glucose to meet their needs. And that can force this hypoglycemia. This bar graph on the right is showing you actually glucose utilization. So the black bar is the LCAD mouse, and it's using more glucose than its wild type litter mate during fasting because of this limited menu. One really important point I want to make is that children with FAODs are at greater risk for hypoketopic hypoglycemia than adults. And there's 2 major reasons why this is the case. First is that small children have a much greater need for energy to begin with. Their metabolic rate is higher. So there was this landmark study that came out in the magazine Science last fall, and they looked at if you just look at this red line, that's total energy expenditure. That's how much calories you're burning in a day. And they looked at thousands of people across all different ages. And what you can see is right at birth, that red line goes way up. And that's the metabolic rate of an infant is very high. And so it spikes up. And this is all relative to their body size. So they basically divided their energy requirements by their body size. And then during childhood, slowly this energy requirement comes back down through adolescence, so that by the time you're a young adult, it kind of flatlines and your energy requirements stay the same. So small children need more energy, and they also have less glycogen in their liver to start with because they're smaller, their livers are smaller, and their metabolic rate is higher, so they're burning through it faster. So in this graph, what you're looking at is the level of ketones in the blood over different lengths of fasting. What I want you just to look at is, like, I drew a line across the level 2, which is sort of like the starting point of a nice ketogenic response. So these are non-FAOD people. What you can see is that a newborn can only go about 2 hours of fasting before their ketones shoot up in the blood. So what this means is by 2 hours, they've already burned through their glycogen stores, and they need to switch to ketones. And this amount of fasting that you can withstand, if you want to use that word, increases quite dramatically through childhood. By the time you're 6 to 8 years old, maybe you could take a 12-hour fast on the glycogen stores that you have. And that's not to say that there's ketones that don't play a role at all during that, but I'm trying to make a point that your glycogen stores improve and get much bigger as you age. So by the time you're an adult, you can withstand a fast or a missed meal much more easily than a child can. So just to summarize what I just talked about regarding ketogenesis, it's an important point that the brain can't use fatty acids for energy. It prefers glucose. If it can't get that, it wants ketones. The fatty acid oxidation pathway in the liver is what provides those ketones by chopping up fatty acids into something that can be made into ketones, which the brain can easily use. And lastly, children are, in general, going to enter ketosis much faster than adults during fasting. And so that FAOD kids are going to be more susceptible to hypoketotic hypoglycemia than adults. So now let's talk about the liver's second trick, which is making sugar or making glucose, which we call gluconeogenesis. So the brain can't survive on ketones alone during fasting. Even during an extended fast, like days of fasting, it was found that the brain, if I could point under where it says starvation, and you see I have an arrow by glucose. What that's showing you is that even under starvation conditions, the brain is getting about 35% of its energy from glucose, and the other 65% from ketones. So you need FAO to make those ketones for the 65%, and you have to have gluconeogenesis to make up the difference. Again, I like biochemical pathways, so I pasted it in there on the right. And here what I want to emphasize is that these two pathways, ketogenesis and gluconeogenesis, aren't competing for the same starting material. So for ketones, you're chopping up fatty acids to make ketones, but for gluconeogenesis, you're starting with a different set of starting materials, usually lactate and amino acids, which come from the protein, often the protein in your muscles. So you might hear people say, if they're losing weight or they're starving, that your muscle mass decreases. And a lot of those amino acids are going to the liver to make glucose. So it's important that these two processes aren't competing for the same material so they can operate at the same time. But one thing they are competing for is the products of fatty acid oxidation. Ketogenesis takes the little bits and pieces from chopping up the fatty acids. Gluconeogenesis takes the energy. So whenever you make something, you have to put in energy to make it. Every time you make a glucose, it takes 6 ATP, and that's what we call the energy currency of the cell, ATP. It takes 6 ATP for each glucose you make. And in the starved or fasted state, that ATP is coming almost exclusively from the fatty acid oxidation pathway. Such that, again, if you have a limited ability to break down fatty acids in the liver, you're going to have limited ATP, and you're going to restrict your ability to make this glucose. Going back to our mice, again, to illustrate this point, these graphs are showing the ability to make glucose, the rate at which the liver can make sugar. The LCAD mouse and the MCAD mouse, these mice still retain some capacity to break down fatty acids and make energy. And they have a much milder restriction on gluconeogenesis than the CPT2 mouse that I told you about that has a virtual 100% block of fatty acid oxidation in the liver. So again, now what you've done is you've restricted your menu, as I mentioned before, by taking fatty acids and ketones off the menu. You're forced an increased reliance on glucose for energy. And you forcing the liver to make that glucose, but the liver is not that great at doing so when fatty acid oxidation is impaired. And that sets up a situation in which your blood glucose can drop. There's sort of this perfect storm of some sort of metabolic stress. Sometimes it's multiple stressors at the same time, mixed in with an FAOD, which is going to limit your ability to make ketones and glucose. And that causes this metabolic inflexibility, is a good way to think of it, where the menu becomes so small, all you have left is glucose. And so your blood sugar levels can drop. So now I want to spend maybe the last 10 minutes talking about something different. Some of the questions that I got from Stephanie about the liver were about immunity. And so I just want to talk about this emerging concept in the field of metabolism in general and liver research, that the liver is an immune organ. And then we'll talk a little bit about inflammation and FAODs. First, I want to define the things I'm talking about. When I talk about the immune system, this is the system that protects you against invaders, bacteria, viruses, toxins. And the immune system, there's several different specialized cells in your body that fight these invaders. And we often think of these as being in your blood. They're circulating around looking for invaders. But it also turns out that some of them live inside your major organs. So in your liver, for example, about 15% of the mass of your liver is a special type of cell called the Kupffer cell, named after the scientist that discovered these decades ago. And Kupffer cells are essentially immune cells. They're macrophages, a type of immune cell. And they just live permanently in your liver, so we call them resident macrophages. When we talk about inflammation, that can be kind of a vague term. What are we talking about? And that's usually the action that your immune system is taking to fight the invader causes this inflammation. Kind of the easiest example would be you get stuck in the finger with a thorn or something. That something is probably dirty, it has bacteria on it. Those bacteria are now inside of you. The cells that are exposed to that send out some chemical signals for help. Immune cells that are flowing through your bloodstream hear that signal, and they actually will leave the bloodstream. They'll push their way into the tissue to go and help. And so now what you have is all these cells rushing to the site of the injury, and that causes swelling, redness, it can cause pain. But what they're trying to do is something good. They're trying to swallow up those bacteria and remove all of the debris from the injury, and so that you can heal again. And one of these chemical signals that cells release are a group of proteins called cytokines. Cytokines are little secreted proteins that can have a myriad of effects on other immune cells, but also on your normal cells, such as in the liver. We call the working cells are called hepatocytes. And so what can happen is these cytokines can signal changes in your other cells and change their metabolism and their function. And just to kind of illustrate as an example of how this might work, I bring up a research study that was done by Peter McGuire's lab. And Peter will be here tomorrow giving a talk. And what Peter's group did was they took flu virus and they put it straight into the lungs of mice. These weren't FAOD mice, they're just wild type mice. And then they followed these mice for several days. And what they saw was, even though the infection is in the lung, your whole body is affected. So these mice became hypoglycemic, which is what that bar graph is showing, because there was a metabolic switch in the body which caused the body to rely more on glucose, similar to what I was talking about with fasting and FAODs earlier. And surprisingly, the liver, even though it wasn't anywhere close to where the virus was also showing signs of inflammation. These Kupffer cells I was telling you about were releasing a whole bunch of these different cytokines that were having effects on their neighbors, which are these hepatocytes, the working cells of the liver. And what Peter showed was that if he put one of these cytokines, which is called TNF-alpha, he put these hepatocytes in a dish and he put on the TNF-alpha, and what he saw in this bar graph in the bottom right was that fatty acid oxidation became inhibited by TNF. So now if you could just imagine, if you say you already have an FAOD and this was happening in your liver, this infection, even though it's not in your liver, it's somewhere else, could be affecting, further suppressing your FAO pathway. And this might be one of the means by which, when you get sick with something like a virus, that you can feel these effects that are like hypoglycemia. You can exacerbate those types of symptoms. And I want to end by just talking about a couple of studies that Jerry was involved in that looked at these cytokines, these danger signals in the blood of patients with various FAODs. Back in 2019, they published a paper that looked at LCHAD and CPT2 patients. And they found some fairly moderate increases in some of these cytokines. A couple of years later, they looked at VLCAD patients. In the VLCAD patients, the more cytokines were increased than in the LCHAD and CPT2. So we don't understand why this difference would exist between the different FAODs. And at this point, we certainly don't understand what these cytokines are doing or even where they're coming from in the body. Is something inflamed? You know, these cytokines were often increased even when the patients felt fine. They didn't have a virus or an injury. So what is inflamed? We talked about inflammation. What's secreting these danger signals? Could it be the muscle? Maybe the muscle is chronically inflamed, even if you're not experiencing active rhabdo. Or another possibility is it could be the liver. There's been a lot of emerging evidence over the last few years in the scientific community that lipid metabolism can play a big role in inflammation and the response to invaders. And one thing that's been shown is that fatty acids can be what we call pro-inflammatory. They can stimulate inflammation. So one possibility is that in your liver, you might be accumulating fatty acids and stimulating these Kupffer cells, these macrophages, sort of making them angry, if you will, so that they're releasing these low levels of these cytokines all the time. So this is an active area of investigation, and hopefully using things like the mouse models and the cell models that I'm talking about, we may be able to get to the bottom of what's going on with these cytokines. I wanted to stop there, and we can take questions.
Did anyone have questions?
Hi, my name is Glenn. My wife and I, we have two CPT2. They're both here, Susan Addison and Luke. My question is specific to the metabolic pathways you kind of referred to. We're always looking at ways to improve our children's experience. They're growing into teenagers and starting sports and so on and so forth. It's about fiber and the body's ability to produce short-chain fatty acids, which you refer to being so important to the heart and the kidney, and that through the digestive process, all the way from gut bacteria through to colon, our bodies do produce short-chain fatty acids if we have adequate fiber in our diet. Can you speak to that, and do you have any additional resources on that?
Actually, I know very little about that. I am aware of some studies that show that, you know, like one of the ketone bodies I showed you, acetone, those things can come from the gut and might indeed be a good energy source for other organs during times of stress. Jerry, do you have any thoughts about that?
A little. So our bodies are bioreactors, right? I mean, they're constantly doing the things that Eric talked about. But for every cell in the body, there's about 1 million bacteria in your gut, and so the body is quite affected by what's going on in the gut. We call that the bacteria in your gut the microbiome. And there's more and more attention being paid to it. For example, in a disease called propionic acidemia or methylmalonic acidemia, if you remember yesterday, I told you that the odd chain fats are important for making the molecule propionate or propionyl-CoA, which then goes into succinate and becomes fuel or one of the components of the TCA cycle. Well, if you have a block, a genetic disorder that doesn't allow the propionate to be metabolized properly, it builds up in the blood and you're toxic. And so there is a treatment that, well, until recently was in clinical trials. Unfortunately, the company went out of business. That they had devised a food. In this case, it was a pill with a special composition of sugars that would promote the growth of the let me back up a second. Bacteria make a lot of propionic acid. And so that was bad for those kids. And this pill that contained a bunch of different sugars that would promote the growth of other bacteria and allow and let them crowd out the ones that made the propionic acid. So it's an example of how the gut does affect your metabolism. Now back to your specific question, the short chain fats can be used in patients with both long chain fatty acid oxidation disorder and MCAD. We don't use them as an oral medication or put them in the formula because for the most part they smell terrible. They're intolerable, and so they haven't gotten a whole lot of attention in the metabolic world. The kinds of experiments that you're talking about have a lot of their origin in studying ruminants, animals that eat grass and digest their food that way. So think cows and goats. It's one of the reasons why goats are smelly. And you can push the gut to make those things. Whether or not we can do it in a high enough level to make a difference, especially when from a therapeutic standpoint, we can give you the equivalent of bucket loads of triheptanone compared to what those bacteria are making. It is the reason why we focus more on what we can give you rather than trying to reprogram what your gut is doing. But it's a perfectly valid way of looking at it, and it would be great to see more research on that. The person who might know even a little bit more about that, Dr. Gilliam, is off with the kids, but maybe we can grab her later on in the day and have a discussion.
One last question.
Hi, I was just curious, as you're talking about the liver, if you could talk a little bit about, as we're talking about teenagers and stuff, and they're thinking about drinking, or they get pulled into that, like how, especially with having an FAOD, like how that changes, because we maybe know a little bit about how alcohol affects the liver if you don't have an FAOD, but if you have an FAOD, what are your thoughts about that?
Yeah, Jerry and I were just talking about that the other day. I was asking him what he thought, and Because when I look in the scientific literature, nobody's done any experiments where they're giving FAOD patients alcohol, or even
Or a
mice.
volunteer just comes to them.
It's something we could do with the mice, but honestly, we never thought to do it. But I do know that just in the liver in general, so non-FAOD, it's known that the more you drink, the more fat gets stored in your liver, because the alcohol actually inhibits fatty acid oxidation. And so let's say you have one drink, but you're probably not so much is going to happen. But I saw a mouse study in which they gave mice a single intoxicating amount of alcohol. So that might be equivalent to, say, having 4 or 5 drinks for a human in a session. And these mice, their livers became very fatty just from that one bout of drinking. And likely it resolves. Like the next day, if they don't drink, over time that fat may leave. But if you already have a risk for developing fatty liver because you can't break down fat, it's probably not a good idea to push that system. Similarly, alcohol causes a big spike in insulin, which can drive down your blood glucose. So if you're already having issues with hypoglycemia, then again, it's probably not a good idea to do that beyond very small amounts.